lecture, so we should have a little fun. Some follow-up stuff. The discussion I've been talking about following up on evolution. Go ahead, your turn.
It's your turn.
Okay. The follow-up on evolution, that's up. I'd like you to finish that before the exam. I don't think it's very much. What it essentially is, we've talked about some of these different concepts in evolution. I more or less listed those from the lecture. So in a way, it's a good review. And I'm asking you to pick one and try to investigate it. And even other ideas that come up, like what has happened in the mitochondria, which I followed up on, but I'm kind of waiting for Ashley to come in and tell her about that, but and the rest of you. But you can follow up on other things too, besides what I've mentioned. But if you go in there, try to pick a topic, and then, then if you're next in line, look to see what other people pick, so we're not all reporting on the same thing. And that way, in a way, you're also making a real nice review for that part of the lecture exam. So, and I don't think it'll take that long, but let's try and knock that out by Wednesday. The review, the genetics quiz is up, and I have that, well, genome, I have that so that it will go into repeat mode when it's turned in after Tuesday, so you guys should be able to see that. And then my next question was that we finished this portion on evolution systematics, and I do have a quiz ready for you, but it would, usually we have a week before the quiz is due, so I can either put the quiz up, we can take it and get some points, or I could just not put it up for points if we don't have time to do it, and just put it on a loop where you can review it. Now there's pluses and minuses to both, and maybe you have a different solution. You maybe want to have it up, finish that quiz by Tuesday, so you have time to go through it as a review also. I'm open to that, but I usually try to give about a week on these assignments, given your schedule. So any thoughts on that, or do you care, or anyone following what I'm saying?
So if you release it for points, we're not going to be able to take it over.
Well, it depends on the due date. I mean, if I release it today and it's due in a week, you're taking an exam a week from today, if I have that date right. So that kind of negates that. So either we move the due date sooner, let's say Tuesday or Wednesday or whatever, or we just make it for, I'll put it up and just, you guys just review for it, just go through it, and it wouldn't be for points if you're going to do it multiple times. So.
Okay.
Yeah, you got an idea?
I was just curious if we repost the quiz, make it due Tuesday, and then put it on the loop.
Yeah, I don't mind that. I think that's great. Gives you the weekend. It's 10 questions. It is a pretty wide pool. There's about 42 questions in that pool. Would be worthwhile going through it. Well, does anyone object to the idea that I mean, it's up and ready to go. I just hit the on button, and it'll go Tuesday, and then look. I've been putting all the review stuff. Like you see, when you put the quizzes back up, it puts columns in your grade book. Everyone gets real confused, but if I give you a heads up, you know where it is. Then look in week 11. I've been putting all the information in there for review, so that then you won't be confused because it's not really an assignment. Okay, so, all right, we'll go with that. Yeah.
Could you put the virus quiz on a loop as well?
It is. It's on right now. Five repeats. Look in week 11, and should be there. And if you're on, you can check it. If not, I'll fix it right away. Yep. Okay. Well, I think that's it for old business. I want to get past this next exam, and then we'll get to the next report. Everyone just kind of gotten their first report in. So if you want to be thinking about that one's more built on interest, past the species level. You can kind of go into a topic on microbiology. It could be maybe a spin-off from your first report and so forth, but I'll try to, I have all the notes written up. I'll try to revise those and get those up, but we'll start thinking about getting serious about that after the test. Maybe I can get the handout out so you can start thinking about it and getting ready if you're really strapped for time. That may help you also. Anything else? Yay. Okay. Well, good. Let's talk about species in bacteria, and I was looking through my slide. I think I've covered a lot of this. Now, we've talked about DNA hybridization. We'll be coming back to a slide on that later today, but we talked about that. Don't be confused. Here we're talking about the whole DNA, the DNA genome and hybridization. Okay, whereas here we're talking about similarity between that one gene, the 16S, if we're just talking about bacteria. So two different types, two different rulers that we're measuring with when we look at the species. And you know, the idea, of course, behind the DNA is how similar or different it is, and that's really a product of mutation. I mean, which is what species are eventually anyway. So it does all make sense by the rules that we know through Darwinian evolution and that kind of thing. So so like I said, these slides are really just a reminder of things we talked about. When we do look at the 16S, we design primers now, they're very well known. I will say, not all primers will cover all the genomes for all the prokaryotes. There are some notes, but you can certainly find what they call universal primers, and you make choices. When we did the Amazon paper, we knew that there's going to be some families that are a little bit more discriminated against than others. And the same way through DNA extraction techniques. You know, one extraction technique just does not pop open all the bacteria, and you go, well, of course, they're so different. Well, yes, and you know, you have to think about it. Some people now with kits just think, well, I just apply the kit, everything's going to work. No, you know, one size does not fit all. But anyway, you can get most of them, and you know, if you know some of the tricks, you can come in with an enzyme that degrades the cell wall, a lysozyme or something else that does that. You get the DNA, you put it in your primers, you know where on the genome they should go. You set up your hybridization technique. Now it's well published, but you still go through it empirically, like we talked about. Okay, you amplify this area, and then you can sequence and see how it plugs in. And the rest of these slides are kind of like that. We've talked about sequencing. This is just really a placeholder to remind you, oh, I've seen that before. I can go back to those areas. And after you get the sequence, you need to align. And this is what I was showing before. I have the other slide from before for continuity, but you'll often get this kind of thing. You go, well, how do you know there's a gap there? Well, it's because you look at the overall alignment, and they'll say, well, there's places here we know are pretty conserved. That's part of what now the general database is. So we can align that based on that, and know that there's some places where some nucleotides have been cut out over time, and other places where they've been inserted and so forth. So that can be all part of it. We don't really care about making a protein here. There is no protein in it. It's a 16S rDNA sequence. So, and what you have here, this is not really showing, I know sometimes people get confused, this really isn't showing hydrogen bonding or double helixing or any of that kind of stuff. It really is just showing alignment here. So these aren't like bonds. Sometimes people get a little mixed up a little bit. And this comes from, I don't know, E. coli and Bacillus, or two different species in that case. So, and same kind of thing. If you start doing this, depends upon the program you use. One, if you are interested in this kind of thing, Clustal is one that you probably will have used or will use in genetics. Still a very good engine for alignment. And it'll help to show you some of the places where there's variability or so, because if you get a lot of these, your brain is still one of the best readers of pattern, but having computers help you where to look is also very helpful in that too. So, we talked about blasting. I think if you're in lab, most of you are. You've done a little bit of blasting. Maybe it was fun. Maybe it's something you want to do more of. But that gave you a little taste of the output, which is just like, with bacteria, it's always like that. But if you blast a mouse, you kind of get a few things. Well, it's really like this species of mouse. It comes down a lot cleaner. With bacteria, you get all these huge readouts, and you get all these similarity numbers that are almost always over the top. And and it can be a little bit overwhelming. And having just a little taste of that, I thought was good for us to do. Even though it's very simple, it still gives us the principle. Okay. Still, taking it to the phylogenetic tree is a good way of going, okay? We've talked about what trees are. They really show relationships. You know, you have branches, so the branches go to nodes. Nodes are where the common ancestor was through evolution. And trees are very different in their making. And I know Dr. Bonnick very big on the interpretation that really was a big part of his PhD, and the phylogenetic work he does with the nautilus is like seminal work. So he's one of the best experts on this, and it comes from a good tradition because the scientists at the American Museum of Natural History, think about it, natural history, it's all about specimens, it's all about classification. That's what those folks do, and they do it really, really well. The leaders in the world. So my point is, going to the fine points of this, you'll get them in other classes. I have a good idea about that you will. Cladistic methods do come up. You define the relationships by examining the changes in nucleotides. That's what we're doing. Use those characteristics that are informative, and then this idea of looking at monophyletic groups when you can. Again, with the bacteria, it becomes kind of a hard thing. We talked about nodes and branches. Yeah, roots. This is if you set up the tree with some kind of ancestor that isn't really part of your investigative group, or oftentimes I just call it an outlier. If I'm just looking at domain bacteria, I'll throw in some kind of thing at the root of the tree, or Aquifex or something, or maybe throw in something like the archaea, which I know is really on the outside, but it's like the benchmark that other things can be compared to, and so the tree takes on more meaning, and you can start adding things like the length of the branch might give you some idea of its similarity and so forth. And that's why this isn't really a rooted tree, it's just a tree showing similarity, but you can go through and work out these similarity indices. There's different algorithms then that work those out for different evolutionary distances. They'll take into account the number and kinds of changes in there. So oftentimes it'll be corrected because you're looking at a subset of a larger set, and you can actually go in once you know more about the model and change some of the parameters, just like when you write statistics. You know, at first you run everything at alpha, 0.05, but you might want to run it at a different acceptance level depending upon the test that you're doing and so forth. But for most of us, we're happy to leave it at defaults and get these things. And then you can kind of say, okay, A and D, have a large evolutionary distance, and they are quite far apart, separated by this node. These guys are more closely related. Their distances are shorter, and really you get usually the same program that'll come in and give you a map, a graph based on this, just like if you had a table of numbers and you're plotting X and Y's and so forth. This is based on similarity and differences. So we're going to talk about all that. This is just showing the difference between an unrooted tree and rooted trees. And now, I'm kind of guessing you've seen a lot of this stuff, so I'm going to go through this fairly rapidly, but these trees are just the way they're showing them. There's really no difference in these, so a lot of times people look at it, well, why is one kind of triangular and the other one's kind of square? It's just the way they map them, and you've probably seen them also now, and maybe I've shown a few in here, but usually these trees are like this, and they just kind of take up like a wall space when you start looking at the prokaryotes. There's just so much variability that it's overwhelming. And now they've even gone to, I think I've shown them in here where they're actually circular, so you can fit in all these little pieces in an area that you can still read, but then you're almost always taking a step back, where if you're studying, let's just say Nautilus is it Nautili or Nautilus is let's say Nautilus is, I don't know, let's just go with that for right now. If I'm grammatically incorrect, I'm sure someone will let me know. But let's just say you have a few species, you know, a tree like this is quite easy, and you pick up a paper and that's that. But if you start doing things like you're looking at assemblages of the human microbiome, you know, almost what you're doing is looking at the amount of detail in a certain region. You're backing off and saying, oh, that's that phylum, and that's that phylum, or that region. So it's like if you map out anything, you know, you start looking at the state of Illinois and you see where the rivers are, but you want to get closer and see the main highways and get into the side highways and so forth. You know, it's almost like that when you get into these thousands and thousands of species that we're trying to figure out in these different microorganisms. And then we also mentioned it, the way to go at the unknown, and there's so much of it with the bacteria, is to put in things that we know. Now this is like standing way back. I mean, we're looking at, you know, just major groups in here with the mitochondria even being shown as part of the proteobacteria, actually an alpha proteobacteria. But you know, you would go in and put in known sequences from organisms that are in the culture collection, so that, you know, if anyone question it or you just need to know it for sure, there is a known specimen, and you start putting these other things in there. You may be able to go in and culture some of the things that you're finding. You might not. There's some fairly creative ways of doing that, so you're not always destroying everything you're looking at. And that is one thing I want to tell you about the Great Plate Count Anomaly. In one sense, we're saying, oh, only 1% of everything out there seems like it can be cultured. And we all go, oh, culturing sucks. You know, and there's a big discrimination in the literature against studies that do culturing. And I'll tell you the opposite. 1% of millions of species have not been adequately described. I mean, we could go out and find new species. We did. We found 100 new species just with one grab sample in Central Park, and are very close to naming 2 things just because it's such a difficult thing. You could go out, and if you put your mind to it, find a microbe, culture it, brand new, name it after you, you know, with the genus name, and do that. That is certainly a conceivable type of thing to do. So that's the opposite flip of the coin of this 1% thing. You know, on one side we say, oh, that 1% is nothing. 1% is still a lot. Even the things you grow, and I can tell you this because I've done this experiment. You can go get a water sample and do a spread plate, and you can probably figure out some of the things that are cultured on there. Probably about a good 5 to 10% of things that you just cultured on any normal agar from the campus pond haven't been described. Okay, I've done that. I haven't had time to publish it, but when people, so, you know, this idea of culturing doesn't show you anything, you know, culturing still shows you a lot. And even if it is only 1%, this is the other thing to talk about. I hope you get appreciation for this. As we keep finding new sequences and putting them in the tree without having anchor points, we're really diluting knowledge. You know, it looks like we're adding stuff, but we're just adding more unknown stuff, and it's hard to know where you're going. Unless people keep trying to discover species and doing that, you're losing your anchor points. You're not gaining definition. Okay, and this is a really big problem I've seen in the world of taxonomy. So I'll move on that, but that is some subtle points I hope you'll think about, and now I think you can think about, because we've talked about a lot of this at sufficient level. 2 tables, once again, just comparing what's similar and different between the 3 domains. Well, of course, the cell structure is different. Nucleus, these kinds of things are pretty good. Introns and exons, that sometimes confuses people, but really the genome for both prokaryotic domains are still rather conserved compared to the eukaryotic genome, which has all this crazy or junk DNA in it. And you can look at a few of these other things in there. they have plasmids, not too many plasmids in eukaryotes, and you start to see them, if anything, probably closer to the protists, things like that. When we get to function, this is the nuts part, okay? I mean, animals and plants do really great things, as do fungi and everything else. When we start looking at some of these functions, that especially, you look at it here, what are all these have to do with? I mean, look at this one you might not know as much, but nitrification, nitrogen fixation, those right there define what? I mean, a big part of what? Metabolism in these organisms, but in ecology, what do you guys study that has to do with this? I mean, you study a lot of things, I know that. That's the nitrogen cycle, okay? You got, anyone heard that? Yeah, I mean, in Illinois, this is like the thing that consumes your natural resources. It consumes this industrial complex that you drive through every day. It's the nitrogen cycle that you're surrounded with. It's the nitrogen that you export, us, in Illinois, that affects part of the world, the Gulf, all kinds of things. The nitrogen cycle is one of the biggest things that people have to deal with. There are whole groups set aside by the Senate and the EPA just to figure out nitrogen in the Midwest. It is a big deal. When you look at these things, where are these processes? Okay, these are two of the very big ones. We've talked about this one a lot, okay? Where are they in? Just for example, well, they're in the bacteria. Okay, now that might not be a surprise to a lot of us, but the nitrogen cycle is dominated by bacteria and is so far described that way. Here is the surprise that has been coming out in the last decade, that as we look into the archaea, these so-called extremophiles, they really are a lot of places, and those processes are in there. And I've gone to Washington several times to review proposals, NSF National Science Foundation proposals, and it's no longer news, but when I did that 5, 6, 7 years ago, the archaea were going, hey, you got to remember, it's down in the archaea. It's kind of a big thing. So when you look through here at these processes, what else do we have? There's similar reduction. This is still sulfur cycle stuff, iron stuff. Rhodopsin-based energy metabolism. Okay, that's light metabolism. Bacterial rhodopsin we'll talk about later today, as opposed to other pigments, carotenoids, and that kind of thing. Chemolithotrophy, gas vesicles done for buoyancy and growth, thermophilic growth. And that all falls within here. So when we start talking about diversity, and you wonder why we're going to spend the next few weeks just talking about this, like a celebration of diversity, the bacteria and the archaea, it's this, okay? It's this. Where it's at, which makes systematics very interesting. Not only can you look at the 16S, DNA hybridization, but you could also pick any one of these functional genes and work out homology and so forth there. So, it's probably a good idea to take some notes on this, because this is already written down. It's really up to you, but I kind of see people looking around at their neighbors like, what's going on? And if you don't understand, you should ask. But this is fairly important stuff. Okay, so we've been talking about genotypic approach to the classification of bacteria. And that certainly is, as we saw with the little video we saw, was starting in the 80s to some extent, certainly taking on traction and teeth in the 1990s, not all that long ago. I mean, compared to Van Leeuwenhoek and those guys, in the 1600s. So that's that's really recent to go ahead and reclassify life on Earth. Just reorganizing your sock drawer, that sometimes seems like enough. Now let's just reclassify everything on Earth. So that was a pretty good approach, really insightful. What did they do with bacteria before then? Okay, Well, they still do this to this day, but the phenotypic analysis is also a big part of that. When I said, what is kind of like the Bible, the book of reference for phenotypic analysis of bacteria, you would all say, hmm? The book, though, that talks about all this, the book that would have where you look up phenotypic, the book where people classified bacteria, even today, but used it in the genomic revolution. What is, we've talked about several times, there's a copy laying on your front desk in 219. Bergey's Manual, yeah, the Bergey's Manual of Systematic Bacteriology. Okay, certainly should know that. Hopefully a lot of you looked at that before you turn in your reports and stuff. It is an important article about how things work. Okay, and what do? It's the gram stain. Is the gram stain just a colorful thing? No, why is the gram stain such an important thing when it has to do with this type of analysis? Connect some dots here. So, yes, it's a stain, it holds in some, it goes in the other. Some are nice and purple, some are pinkish. Why do we care about that? It does help us classify, we care about that, but what is it tied to? Who here has heard about the gram stain? Okay, 5 of you. Okay, the rest of you, look over your notes, go to the lab, pick and look at any of the stains on the table. Okay, you've all, well, I know you've heard about it, whether you remember or not. The gram stain is really, really important. Who can tell me something about the gram stain? We seem to have we're missing a point here in connecting dots, kind of like the log in photosynthesis. So let's connect the dots. So what are the parts of the gram stain? After we make a smear, okay, and we heat fix, now we got some bacteria on a little piece of glass. What do we do next? Go ahead. We add crystal vial, okay, next? What's next up?
Iodine.
Oh, we rinse it, then iodine, yeah, okay. Rinse that. We add alcohol. What's the alcohol doing at this point?
Decolorizing.
Decolorizing, right. What's staying purple? Hmm? Yeah, everything at this point is staying purple. When we destain, we're pulling the purple out of the gram positives or the gram negatives. Okay, so you got this. Now, what's the difference between a gram negative and a gram positive bacterium at this level? Cell wall. How is a cell wall formed? Gene expression. Okay, so it's the phenotype. There's an expression to make that. It just didn't happen. It happens to every E. coli, from whenever they started to wherever they're going to go. Okay, they're going to express that and make a gram-negative cell wall. The gram-positives are going to do that. So it's a phenotypic expression of the genes, and we can measure it. So, and it's reliable. So it's a characteristic. Okay. Other things, shape. Shape is pretty good, but shape. What do we know about shape? It can sometimes change, right? Some of you guys came up against that. Oh, it was a rod yesterday. Yeah, sorry about that. That just changed. So, is it a characteristic? When they were first starting to figure out the genomic type of stuff, I attended seminars on that. This is a characteristic, especially in the gram positives. This is a descriptive. Okay, it's a characteristic that usually works, and that's what the Bergeys will say. It's 75% positive during this time point, then it becomes pleomorphic. So there's a lot of description around it. Okay, we can look at the fact that what's this talking about? Faculty of what? Usually, yeah, respiration. Okay, so that's how you make a living. Okay, so that's a characteristic. And then we can get more and more specific just by looking at these tests. We're looking at an expression that makes the cell wall, that makes shape, which is pretty good, that makes the metabolism, and getting more specific, what things are being metabolized. And we have little chemical tests based on enzymes or responses that tell that. The Bergeys manual sits there with table after table after table on this. And that's how it tells you, it even tells you how reliable that is, because what do we know about phenotypes? We know they're variable. Look around. I mean, we all have the same set of chromosomes, not the same DNA in the chromosomes, we have the same sets, and we kind of see, well, I kind of look like this, and you kind of look like that, and yay, but we're not, don't all look the same. And it's the same thing in bacteria. Some of you came up against that. Wait a minute, why am I getting kind of a pink change here? I really wanted this change. Yeah, I know you really wanted it. There's variation, okay? It shows up. It is how it is. And people realize this, and they record that. So this was a very good way of classifying stuff. We made a lot of headway here. But in most cases, what is this being tied to that really limits us from studying the microbial world?
Being tied to being able to overcome.
Bingo! She said being able to be cultured. Okay, so again, strengths and weaknesses of approaches. Let's see how other ways we can cut stuff up. Okay, I kind of talked about all that. Kind of went out to all that too. Okay, in the cell wall, in the membrane, let's go inside. In the cytoplasmic membrane, as I mentioned, I showed you phosphatidylethanol as being the specific type of phospholipid that the bacterial cell lipid bilayer is made out of. And if you go back to that, you'll see, oh yeah, there's an ethanol group, there's a phosphate group, and there's two fatty acids. What I didn't tell you at that time, just like early on, you learned there's only 3 shapes of bacteria, is that there's more to the story. Phospholipids can vary, as you can see here. I'm not going to ask you to draw these things. Probably if you've had organic, these things look very familiar to you. Organic is no longer a prerequisite for our course, so it may not. You may be going like, oh, that's kind of nice. I wish I had a T-shirt that looked like that. Okay, but what we can see is some things that we do all have some idea from, and that is some of those fatty acids can be saturated. They can be, if you're saturated, you're saturated. You're unsaturated, you can be polysaturated. Depends on where you're saturated at, and if there's other groups, how the fatty. Now you can, once you're saturated, you stop the turning around the bonds, so that cis and trans and some of those things become important. Okay, if we have cyclic nature of some of those, and branched and so forth, if we hydroxylize some of the fatty acids, they're different, and they are different. Okay, they are. Why? I'm not exactly sure. I don't know how it lines up as far as phylogenetics goes, but I can tell you what people do with this. You can grow up a culture. Again, it's culture dependent. We've done this. We've done these analyses in several of our papers. It's very reliable. Grow up your culture. You extract the lipids. Okay, it's fairly easy to extract lipids, as you might imagine. You use some like a detergent or something, and then you make an ester out of that, and you take that ester and you run it through. I think a GC, would that do it? Or what would analyze fatty acid esters? HPLC, I think it is. The HPLC, and then gas granulate, GC. Okay, so you go through a GC and you read these peaks. I mean, it looks like that. What you get from that then is a pattern. And here now you got to go back to a database. People have gone through. They've done this with E. coli. The peaks look like this in the database, and then you saw in the database. Okay, and then you take your bacteria that you've cultured. You do this, and you get a % similarity. That's it. You don't get, you have E. coli. Very rarely. Even if you put E. coli through there, sometimes you don't even get E. coli. It's just a similarity index, but it's very close to that. So you can help to identify organisms. When you try to name a species, they ask you to do the FAME analysis and put that in as evidence along with everything else. Why? Because people are going to be, you're building a database and you're building benchmarks. You want to have as much evidence for that benchmark. You're building a case, you know. If you're building a case, a crime case, you want to get as much evidence to make it solid as possible, okay? So in this one, what's the advantage? It's not that hard to do, and it's not terribly expensive, so it's relatively rapid. What do you think a disadvantage is in this? If we were to study the human microbiome or the microbes of the soil and the cornfield out here, what might be a disadvantage of this that you could think of? It's okay, anyone can say anything. I welcome. What do you think is a disadvantage? Oh, that may affect what you're looking at, but the big advantage or disadvantage here is this part, again, okay? Right here in front of you, okay? This type of database tends to be really good, first off, with clinical serotypes and clinical species, because they've grown in somebody, or something, so it's really strong that way. It's not as strong when you start going out into environmental samples or other samples where you can't grow things. So that's one thing. Okay, this is DNA hybridization. This is kind of the gold standard, and I have talked about it, so I don't feel like I need to go into it a lot, but this is a nice way of showing it. If you had organism one here that you're trying to compare things to, you could go through and label it, P32 or some other kind of label that would be the same. And you have these other organisms. In this slide, it shows you if you took that same DNA and denature it and hybridize it to other DNA, either itself or to other DNA of that same organism, it's going to be 100%. Species 2 is pretty closely related to species 1. A lot of the DNA is shown that hybridizes. You can look at the percent label that comes through after you do the hybridization, but some of it just doesn't. Okay, this one has less so, this one's even less so. So if this is happening to the degree of what percent, at what percent DNA hybridization do I have with different species? Or 70%. If you have 70% hybridization, it's still considered to be in the same species. It gets less than that, and you're going, well, different species, less than that, different genera, and you kind of work your way back. Okay. What's good about this? It's very specific. Again, some of the same seminars I attended, this is still kind of the gold standard because you're getting at genome to genome types of questions. The disadvantage is, again, you need a lot of DNA to do this. You just can't pick up a cell and do it. Now with genomic, we can amplify maybe the whole genome. It might come back again. But why do it if you have the whole genome and you sequenced it? You can just do it with a computer. You don't have to do it with phosphorus. So that's what people are arguing right now. If we can do genomes so fast, let's just sequence them. That's what you'll be seeing, in your lifetime a lot more. So that just goes into, above 70, now 25, it's showing you that kind of cut on. This is just saying that when people are looking at different strains, looking at different alleles, comparing other aspects of both species becomes important. I'm not going to dwell on this too much on these, but like we mentioned with the other genes, you can look at something else besides the 16S. You can look at changes in other functional genes, and sometimes that's important because functional genes have a lot more constraint on them. If you change a gene that is leading to a protein and the protein no longer works, that species quite likely goes away. So there's a lot of advantage to take a multi-gene approach, and now that we can do multi-genes, it's a lot more appropriate. Okay, GC content, this is guanine cytosine, and we'll see this a little bit when we talk about the gram-positives in here, but I'll introduce it here. This is talking about the amount of G and C's in the genome. And it's interesting, because, we have 3 hydrogen bonds here. There's some constraints in some organisms that as they go into different temperatures that the GC content will change. But anyway, they show here the relative amounts of it. It'd be hard to set up a whole phylogenetic tree with just looking at G's and C's. But we do see some whole groups that have an amazingly high G and C content. So this will come up in some of the microbial systematics. I throw it out here for your consideration. You can see the range of things in that. This is just a reminder, we've talked about this one. This is the tech collection here. These are the guys in Germany. They're very stringent, I found out. We could not ever get our DNA hybridization to work with these guys, so probably our fault. Let's just say it's our fault. But there's the Japanese have all these are great collections. I'm most familiar with this one, and this one. But if you make a species or you find a species, you have to deposit it into 2 internationally known repositories, and that would be that. What I'd like to do in here is start talking about microbial diversity. I don't exactly like the way the book dealt with this. It used to be that they organized it and they went phylum by phylum, and that was really great. And then in this edition, they took some of that out of there and they moved it into this functional diversity aspect, which I like at some point, but I think in teaching it may confuse you. So I want to start out with a preface on this, is that we will, in this first part, focus on function and who's doing what. We're not going to worry as much about how it fits into the phylum. I will be mentioning some of the phylum names only because next we're going to kind of do that dance right through all the different phyla. So I thought about just going completely away from the way that the book does it, but there's so much detail, I think that would even be more dissatisfying, so we'll do that. I started with kind of the Star Wars idea. This is actually, you've done DAPI, and you can kind of see, you can look into it, it does look kind of like stars, but what we want to talk about here are the major groups, and we're going to look mainly in this first part at the domain bacteria, and what I'd like you to do is to focus on key characteristics, okay? And that's what I'm going to hit on. Look at this, look at that, and then, you're going to say, wow, that's a lot to know. It is a lot to know, but at some point you had to learn some names of plants, and you had to learn that the squirrels are different than the foxes and stuff, and you have not a lot to go on here. Unless you've been sick from a different microbe or something, you probably really haven't paid too much attention to it. So it's going to be a compromise, and I realize it's a lot of stuff, so I tend to go with like matching on these. That is, here are some names, here are some characteristics, but usually many characteristics. So like, oh, I forgot that one, but yeah, this has got to be this one. That's the way I usually go on this kind of stuff. But I will tell you straight up, start taking, I mean, you got to the slides, but take a few notes, make a few flashcards, whatever it takes on that. And I will try to be parsimonious as much as I can. Diversity, again, we've talked about a lot of this. Metabolism makes them diverse. We have organisms that are the nutrient cycle. We have things that can metabolize rock. We also have things that can live not only as parasites and therefore not really have to deal with what other microorganisms have to deal with in nature. Okay. But then we have some that just have these amazing adaptations. This is Deinococcus radiodurans. Okay, it's not that hard to find. I have a culture of it in the deep freeze if you want to play with it sometime. It itself is not radioactive, but what's interesting about this thing we'll come to find out, A, it's related to Tachyquaticus, or Thermus aquaticus. Well, why? It's just where it falls out. But it can take more radiation than any other organism that we know about, and still be fine. So people are really interested, well, how does it do that? It has a waxy cell wall, but it can repair its DNA. Oh, well, can we learn from that and maybe repair our DNA or something? So its genome was done a long time ago. People are still trying to figure it out. So we have things like that to talk about. And then you're going to find out in here that we will talk about a lot of variation on form, variation on reproduction. It's I think, pretty interesting. I have to go back to E.O. Wilson. I hold him up because when you talk about diversity, you should talk about E.O. Wilson. Here he is, looking for ants. Okay, he's a sociobiologist, so and if you see him, it's not unusual. He's walking around looking at ants, and here's this Harvard professor that looks, to some people undignified. I think he's being a scientist. Okay, here's a book I mentioned here. If you get a chance, I say read it. You guys are fast readers. I think if you like science, so forth. I love this picture because until you read the book, you don't realize one of his eyes don't work. He spends most of his life looking at small things. So here's a person who's overcome a handicap to begin with. And of course, I like this, okay? If I could do his life all over again and relive my vision in the 21st century, I would become a microbial ecologist, yay! 10 billion bacteria in a gram of soil, about what you hold between your thumb and forefinger, thousands of species yet to know about, and it goes on to say, I'd go with my microscope and new techniques and look at that. The point is, he recognized early on, this is written, I saw him at Kent State, so that was in the late 90s, and he said, oh, and he signs his book, and he makes like a little ant, like as fast as you can imagine. And he says, he's a very personal guy. He says, well, and he just gave this lecture where he ended with this quote, and he says, so what are you doing here at Kent State? He said, oh, I'm a postdoc. I'm learning about microbial ecology. And he says, the world is open to you as a scientist. And I go, oh, thank you, Dr. Wilson. So fun. But anyway, I like this because of the diversity, and it's recognized at a real large scientific level. So let's start talking about it. We're going to start banging our way around the eubacteria, the bacteria, and this phylogenetic tree. And we're going to start out in this chapter looking at phototrophic, chemolithotrophic, and methanotrophic proteobacteria. So going back, we're looking in then the proteobacteria, and I just want to tell you now, I'm not going to be asking you so much to know that there's 5 and I think now 7 different subphyla of the proteobacteria, but that we're going to be kicking around in this new, probably a new word for all of us, the purple or proteobacteria. Okay. Phototrophic, what kind of bacteria do you think we're going to talk about here? Oh, you're quite fun. Okay, chemolithotrophic. What kind of bacteria do you think we're going to talk about there? Okay, methanotrophic. What kind of bacteria do you think we're going to talk about there?
Bacteria that use methane.
There you go. Okay, so we got methane. Yes, methane eaters. Okay, we don't see when you're talking about producing methane. If you've ever gone into a swamp and you see those bubbles coming up, yes, they smell like hydrogen sulfide, but you can walk around with a garbage bag or something and capture those bubbles, and put them in a bag and have someone push the gas out and light it with a lighter. It won't blow up, but you'll get a massive flame. That's methane. Okay, that's methane from the bowels of the earth. Okay, those are not. That methane is made by prokaryotes, but the methane is made by methanogens. Those that have a genesis of methane. Those are archaea. but there's also bacteria that eat methane. And it's really, really interesting, I think, to know that we have a lot of things that will do a C1 metabolism, something you don't talk about, maybe not that much. Okay, chemolithotrophs, what do you think? What do you think those words mean? Trophic, okay, to eat, get it? Okay, at least I'm gonna get energy from something there. Chemo, chemicals, what's that litho thing about? Hmm? Just say it louder, maybe saying the right thing. Inorganic, yeah, litho, rock base, no, but inorganic, okay. Phototrophic, you probably all know it, you're talking something about with light, so we'll get into it. The chapter and this starts out with the prochlorophytes. The prochlorophytes are essentially really, really closely related to the cyanobacteria. It's probably the prochlorophytes that were the organisms that went into endosymbiosis to establish the plant chloroplast. Okay. Either way, if you remember cyanobacteria and their closely related cousins, the chlorophytes, that's fine. Okay. We've talked about the cyanobacteria in this group, and since we're talking about functional diversity, we talked about nitrogen fixation. I think that was only a few days ago, so I won't go too much into it, because I don't want to hold up your learning in any way, but realize that with the cyanobacteria, nitrogen fixation happens in a specialized type of cell. Okay, that cell is called a heterocyst. What's cool about the heterocyst is it does have nitrogen reductase, okay, can reduce nitrogen from the air into an amine group, and oftentimes the glutamine. Now this cartoon shows like there's a big channel, like I-94 running through these cells. It's not quite that way, and let me tell you why. This reaction to reduce, most of your reducing reactions have to take place under reducing circumstances, which usually means reduced oxygen or low oxygen, okay, very low amount of it. And thus, in cells, the vegetative cells that are producing oxygen, you can't get that to work very effectively, actually not at all. So the special cell type is put in here that reduces the amount of oxygen that can permeate into it. Now, some students go so far and think, well, it just has to block all the oxygen out. And if you think about that, that would be the best, wouldn't it, to have nitrogen reduction take place when we do it in a test tube. That's a great way to do it. Some of you tested for nitrogen reduction, and it was in a fermentation tube after oxygen went away. Okay, so you get a fuel for that. But what would happen if the heterocyst was just like a little cellophane bag and there was no gas exchange? That's right. You would not be able to diffuse in nitrogen gas. Okay, so there has to be some kind of permeability. The other thing is once you've reduced it, you do have to have some kind of way of getting that nitrogen, glutamine, an amino acid, if you're not familiar with that term, to go in and become part of the cell growth. So there is exchange, all right? But this little diagram on the functional aspect of cyanobacteria that we're talking about also works for some of you who are going to start looking at nitrogen fixers in the roots of plants. Plants, nitrogen fixation in legumes takes place where? Botanists and ecologists and people who garden. And little nodules, okay, if you pick up an alfalfa plant or a soybean, they look like they got little things growing on, right? Those nodules are not just ugly little things to gross you out. Those are actually areas that can reduce oxygen and do the same kind of thing, only on a much more macroscopic level, okay? It turns out, I don't know if we'll cover too much in this class, but it turns out that when a legume wants to produce a nodule, it'll actually exude quinones and different chemicals that will specifically invite the nitrogen fixating bacteria to come up into there. Also, they will send out the little signals to tell root hairs to curl and then to kind of globulate and make the nodule in this little home form. So it's a very intricate symbiotism that goes on with plants and bacteria in that, but it's for the same function, the same game. First seen with the cyanobacteria, and that's a function we wanted to talk about here. So, we've talked about gas vesicles, we've talked about some of these other things, so let's move on. It just keeps going. So, type 1 and type 2 photosynthesis, all fix carbon dioxide and carbon as a carbon source. Some use glucose and acetate at night. That's part of photosynthesis, okay, but the cyanobacteria will do that also. So they can be photoheterotrophic at night, realizing, I think we all realize photosynthesis doesn't occur without light. Okay. Some people always say, oh, that's a cyanobacteria. That fixes nitrogen, because they learn that in high school or something. Most, not all. Okay, just because you're a cyanobacteria, there's different lineages. So I like to talk about that, and so forth. Yeah, some can actually, if there's sulfur around, change their metabolism and deal with sulfur and both reduce it and or use it as an electron acceptor if it's in the sulfate form. Cyanobacteria are very versatile that way as a group. And I'd love to talk to you more about that. We've seen that already. The prochlorophytes have chlorophyll A and chlorophyll B. Probably aware that there's many different types. They have phycobilins, this accessory pigment. And this is where people start to match this up. How did this really come about? How does it really work in plants? And it turns out that photosystem 1 did evolve with bacteria, cyclic photosynthesis as opposed to linear photosynthesis. That's pretty much where it ended. Now let's look at the prochlorophytes. They do resemble cyanobacteria. They have chlorophyll A as the cyanobacteria did, but they also resemble the green algae in the plants because they have chlorophyll B. So people are thinking, this might be where some of this symbiosis happened as opposed to the cyanobacteria. How would you account for that other pigment is the argument on this. So that's it. Okay, we're going to move to the next one. We've already touched on some of these in our other talks, so I won't spend too much time, but now we're into the purple phototrophic bacteria. This is what we said we'd be talking about. These guys have a purple, they reflect purple light, so they're photoautotrophic, and these are the ones when we talked about in evolution that were the anoxygenic organisms. And I think this is really reviewed, so we can look and at really the slides I used before. We're substituting hydrogen sulfide for water. Oh, what? Light moved up. Of course, light is in waves. It can go anywhere it wants to. So anyway, we're converting the sulfide to a more oxidized state, okay? And we're doing that using energy from the sun, only certain wavelengths of that. And so it also is an autotroph. So these would be actually autotrophic bacteria. And it's like very much like photosynthesis, but it happened before things really got going on the water and so forth. Water wasn't as free of a molecule at that time. Now, let's take that process, and now we've seen more or less twice, and look at the world. Now, this is kind of interesting. The one to remember here, I'm going to try to highlight certain genera for you. This is Chromatium, okay? Chroma, I don't know. I always just try to relate that somehow to purple in my mind, but Chromatium, okay? Anoxicurious of lakes. The best one with Chromatium, we have Thiospirillum here and Diophidia. Chromatium is the one I want you to look at. If we were to open up the Winografsky column and go into some of the purple areas, it's quite likely, this is a light microscopy picture, that we would see bacteria that look like this. what are those nodules? Are those nuclei? Are those mitochondria? Maybe it's on the slide, maybe you're reading them. Oh, no, Mike, read the slide, I don't know.
So,
It's sulfur, Bob, is it on there? Yeah, sulfur inside, yeah, there you go. Ding, ding, ding. So this is sulfur, why are they doing it? Remember the inclusion body stuff we talked about? Why do they have sulfur in there? They took sulfide, okay, I'm gonna grab the electron, or I'm gonna grab the electrons off the hydrogen, okay, and I'm gonna, in the meantime, I'm gonna oxidize this sulfide, to sulfur. Why are they hanging on to it? It doesn't seem like there's a lot of room in there. For a rainy day, can you explain more? What kind of rainy day are they trying to get ready for? Yeah, they have a sulfur and they could oxidize it further in this case, so they can do it. They probably won't move it all the way to sulfate in this case. I don't think that chromium does, I have to check that. But they keep it as an inclusion body. So this is something that we can, is it weird? No, we found this in sand, we find this in our gardens, we can find this all over the place. And you say, well, why don't I just walk outside and see purple earth everywhere? Because remember, in the Winografsky column, we're taking these conditions that probably are happening within an aggregate of soil or a layer of soil. It really depends upon how the redox potential is worked out. And we're taking that habitat and we're opening up that window so we can see it. That's why we just don't walk around and see purple all the time. Although if you look closely, sometimes this time of year, when plants are decaying, and you look at the base around things like spartina and so forth, you'll see purple bands and stuff. Sometimes you can get a peek at some of these. They have to be close enough to the light and conditions. You'll see this in nature a little bit more. These guys are cool. They got inclusion bodies too. This is usually the one we see the most often. So that's why I say take a look at chromatium on that one. Okay, purple phototrophic ones. We're still in that group. There are some that have a basis of sulfur, and that's the ones we talked about so far because we're talking about how Earth became Earth, right, with life on Earth. Some actually will use organic compounds, some will use elemental hydrogen, so they can get their hydrogen, their electrons from other stuff. Okay, so this group has a little bit, this is a subgroup, this group has a different way of going at that, is the only point. So we have purple sulfurs, purple non-sulfurs. Photoheterotrophy as opposed to photoautotrophy, and that should mean something to you in this case also, and I think we can move on from that. Here's a genus associated with this, Rhodobacter. Okay, Rhodobacter, probably when we, again, if you open up the Winografsky column, probably Rhodobacter and Rhodospirillum are the ones that we most likely see, and they're a little bit harder to pick out. I mean, you put this around a light microscope, even this is at probably using, phase, it kind of just looks like, little dots. So it'd be harder to pick that out. Chromatium kind of sticks out a little bit more. So where would you find it? Exactly what we're talking about, places that are anaerobic, that would, but you can still get close enough to light. You can get that light to come in, that's a big deal, something that Winografsky figured out. Here's my favorite. This is the genus I'd like you to remember, Rhodospirillum. Now here, almost spirochetes or spirals. Yeah, no way they are. Okay. Rhodospirillum is a true spiral. This is that third shape we always have learned about. Okay. They look like this. They look like spirals. If you open up a Winografsky column and look at these guys, and you look at them under phase, you don't kill them. So they'll come through, and they're big. I mean, you're used to looking at E. coli. Oh yeah, a little bit E. coli. These guys are bigger than E. coli. They'll kind of move through. They kind of roll through like big Cadillacs through the microscope slide. You can't really see the color with white light as you can when they get together and they reflect the light. Okay. So Rhodospirillum, purple non-sulfurs in that group. Chromatium is a good one for the other group. We pretty much know about this, the Winografsky column. Here's one. These guys have plants on the top and they have in a jar. Did I make the bet with you guys on this, the $50 bet? Now you're listening, aren't you? Okay, no. What I did with the advanced guys a lot, you see this? This is a voltmeter. Okay, it says 523, okay, that's millivolts. That's potentially a half volt of electricity. That's true. Now, chemists in the room are saying, yeah, but you put sulfur down here and some carbonate, you're just setting up a big kind of battery, aren't you? Well, I got Keenan thinking about this, and he went in and set up some of these with his daughter, and they hit some with antibiotics and some without it. When they hit with antibiotics, the voltage dropped. So it's a biotic response that sets up between the actual overall transfer of electrons. Oh, what's going on here? What's going on here is, I don't know which is which, but when we set it up, this is a wire that has insulation. We tear off like the last inch down here, so it's the bottom part of the current. And up here, so it's all insulated, and on the other end we have a wire that's stuck in here, so it's measuring this potential. So I used to argue with my students, if they can get a light bulb to go, I'll give them $50 off of a Winnigrassi column. And then they start saying, well, how big of a light bulb? Because, amperage, right? So they started to get those little tiny light bulbs, they couldn't do it. But you know what we haven't done it yet, but you know what the answer is. Then they start putting a whole bunch of Winnigrassi columns. You gotta, it's series versus parallel circuitry. Should do it, but I still have the $50 bet. If you guys can do it, I'll give you $50. I would just love to see it. So, but it potentially should go. Okay, so you're seeing the different zones in there. That's a little bit of an aside. We see in here though the places where we're going to talk about the sulfur maybe today. Here's our rhodiosporidium in that area, chromatium. We don't talk too much about beige jatoa for us, but these guys are found like in the sewage plant effluent. They call it sewage fungus, but they're just really long filamentous bacteria. And we'll get some of those guys in a little bit also. So. Anyway, talked about that. That's kind of fun. One thing you probably realize is, from studying ecology, the deeper you go down into sediment, it gets more and more anaerobic and so forth. And the redox, the millivolt potential, when it's oxygenated, is like plus 200. When you get down into deeper areas, it's minus 400. And that difference in potential dictates which terminal electron acceptor is used by the microbes. So just one way of relating maybe what you're learning in your other classes in soils to what's going on in the microbes. So, there it is. I'll give you $50 if you light up a light bulb. Okay. I'd love to have that on visit day. I go to these preview days and it's like, oh man, it'd be nice if we had like a little mud and a light bulb. What's going on here? I don't know if people would like that. Okay. Green sulfur bacteria, these guys are sulfur bacteria also. They are not, they're in the functional area of dealing with using sulfide, okay, and they're photoheterotrophic, and they will make some kind of inclusion body also. Here we have, at least initially, okay, but we have so these are green, so think of chlorophyll. Okay, so chlorobium, and chlorochromatium. Oh, chromatium was that other one that dealt with sulfur. This is the green version. That's kind of how that one is started. Yeah, even though they will metabolize in these chromosomes, these guys are different. These are the guys that made the big sulfur deposits in parts of the world. They would metabolize, they would oxidize the sulfur, you get elemental, and you can see this in the Winnigrassi. Have anyone noticed in the Winnigrassi column the yellow-green deposits? Look in there. You're getting elemental sulfur, and they're coming from these guys. They put the sulfur on the outside. You can actually see elemental sulfur being produced, which, when you see it in the jar in chemistry class, it's really bright yellow and so forth, but really in this phase, it's kind of greenish yellow, and it is from these guys, so that's kind of interesting. So these are green sulfur. They're real, very similar to the purple sulfurs, but that sulfur goes on the outside for these guys. Not gonna tell about those guys. Let's start the sulfur cycle, yeah? You up for that one? Okay, yeah, see. Good. All right, here is an interesting vibrio. You know, vibrio is like a spirulina, but you can look at, it's kind of a comma-shaped kind of guy. You know, when we talk about Vibrio cholera, it's gonna be that kind of comma thing, a kidney-shaped looking bacteria. That's a vibrio, all right. We're gonna talk about sulfate reduction and disulfovibrio in this one. So even though we're talking about, we're still in the proteobacteria as a group, that's great. Don't worry about group 17. Yeah. But we're gonna talk about sulfur reduction. So now let's get our heads around that. What kind of sulfur, what redox form of, what oxidation state of sulfur can we use to reduce? Is sulfide gonna work for us now? Can you reduce sulfide? Are you in chemistry? Can you reduce sulfide? No, you really can't. And you're right. Okay. What's a good candidate? Sulfate, as we have a sulfate and sulfur, great to reduce. Sulfur's in the middle. It can go, either way. It's, I can get reduced, I can get oxidized. So it's in there, and it works really good. Sulfate's the one we're going to reduce. Now, this word, we've talked about, but maybe not in this class. Dissimilative versus assimilative. Okay, I want to get this down. When we think about bacteria in nature and in us, some of the things that they're doing, they're used to assimilate. For instance, they can assimilate parts of proteins or amino acids, and they make them into other proteins or enzymes. Okay, dissimilative just means that it's used in metabolism, but it isn't assimilated into their cell. Okay, in this case, it's going to be a little electronegative molecule that can accept, it's going to be a terminal electron acceptor. So it's, in a way, you can think of it like, oxygen and respiration. We need it, we don't really assimilate the oxygen directly in that pathway. We may assimilate oxygen into other pathways and so forth. But as a terminal electron acceptor, it is a dissimilative. Okay, disulfovibrio, basically we were talking about desulfating the sulfate in that idea. Sulfate is electron acceptor. Okay, kind of talked about all this. And we talked about the similar. So that's all there for you to look at. One thing to think about, as though you don't have enough, but I'd like you to think about the nutrient cycles. Now this is the sulfur cycle in a very simplistic state. Sulfur cycle is still pretty much dominated by microbes moving sulfur around in different states. The nitrogen cycle is certainly that. In nutrient cycles, you've all done this, right? In general biology or ecology, at least talked about carbon cycling, nutrient cycling. Yes? Anyone not do that? Okay, so you all have. Good. Then you realize that when you look into a nutrient cycle, they're partitioned into the oxidative parts and the reductive parts. This has to make you think about one other thing. In the course of evolution, there probably were not nutrient cycles for about the first 2 billion years. Okay, what happened to all that stuff? It's something perhaps to contemplate. So, with the sulfovibrio, they are reducing things, so they do this best in a reducing atmosphere. So they are in, they're obligate aerobes, okay. Well, this is an aside. Some can also use nitrate as their TEA because nitrate is so much like sulfate, okay. The one thing I want to show, so in here where are we at? We're taking sulfate and reducing it. Oh yeah, so we're coming across, we're doing this shortcut across this nutrient cycle. Last slide here is just to tell you that it's true that you can use, they can reduce sulfate. They can also instead of using hydrogens, they're able to substitute hydrogens on the PCBs, taking off the chlorine. So they do reductive dechlorination also. So the sulfovibrio and some of the methanogens are able to take apart PCBs, some of the worst carcinogens ever made by, primarily by Monsanto of St. Louis. Okay, we'll see you guys, have a good weekend, see you on Tuesday, and what am I doing?